Hi, everybody. Good morning. This lecture is on biomolecules. And so, what we did the last lecture with chemistry is we gave you kind of a foundational understanding of how atoms interact to form molecules, and then how those molecules have personalities and identities. And we also covered functional groups and how functional groups can change the personality of a of a molecule. Um, and so what we're going to jump into next is we're going to kind of look at some of the principles that we learned from those molecules, um, and and use those principles to kind of build and develop a greater understanding of these molecules, which are used in all kinds of biological systems. So every single biological thing, anything that's living, right? Any living cell is going to have these four components inside of it. Every living cell will come from these four things. Okay, so moving forward, the first one we're going to talk about is lipids. And so if you look on a nutrition label, this will be the first thing listed. This is fats, right? And fats are just like anything else. Any of these other molecules, they have a monomer form. So there's like a basic structure. And then that monomer form can be duplicated and put into larger structures into a polymer form or a multiple form. And so what we're going to do is look at the basic structure of a lipid first, okay? So the monomer of a lipid is called a fatty acid. And there's a reason why. Fatty acid, there's two parts to a fatty acid, and you'll see that in a second. And there are two different types of fatty acid, right? One is going to be saturated, the other is going to be unsaturated. So the monomer of a lipid is a fatty acid. One is saturated and the other is unsaturated, right? And this makes sense because you've seen this before on like a nutrition label, where it says total fat, and then it gives you some grams of total fat, and then it says how much of that total fat is saturated, trans fat, or unsaturated. And you might say, like, hold on, trans fat, that's something else, right? No, a trans fat is just like, it's a different type of unsaturated fat. Okay, so what makes something saturated or unsaturated? We'll cover that in a second, but this is the basic structure. This particular one is saturated. And the way that I know that is because every carbon atom in this molecule, besides this one colored in red at the end, every carbon atom in this molecule is saturated, its bonds are saturated by hydrogens. There's no double bonds down this tail at all. It's all just hydrogens surrounding carbons. Now, if you remember our lecture about functional groups, what you see here is a methyl group. And we know methyl comes from the word like methane, right? Methane being a carbon bonded to four hydrogens just floating around. It's a gas, it's a small molecule, but it's flammable, it's combustible, has a lot of free energy in it. This molecule right here is also flammable and combustible and has a lot of free energy. Like gasoline in your gasoline tank is made up of molecules like this, right? They're hydrocarbons. They're fossil fuels, right? So, anyways, this fatty acid has two parts. There's a hydrocarbon tail, and there's a carboxylic acid head. So this is our carboxyl group, right, over here on the left. This is our carboxyl group. I'm going to go ahead and point her out. Oh, there we go. This is our carboxyl group. I didn't even know I had this feature. Yee! There's a carboxyl group over here. Sometimes this hydrogen here that's bonded to the oxygen will pop off, and it will just be a proton floating around in solution. Whereas this oxygen is going to hold on to the electron it gets from hydrogen, and that's what makes this an acid. Is that this hydrogen proton can pop off in solution. So there's a carboxylic head acid, and then there's a hydrocarbon tail, and this hydrocarbon tail is full of free energy. It fuels our automobiles, right? It fuels jet engines. It fuels the Apollo rockets. So, anyways, this is a saturated fat. We'll talk about those in a second here, right? Just ignore these boxes. I had to modify this image just a little bit. Saturated fats. So a saturated fat has all four bonds of each carbon saturated with hydrogen. Now, that's partially true, right? If you have a carbon bonded to a carbon, bonded to a carbon, bonded to a carbon, two of their bonds are going to be taken up by other carbons. But it means like every bond that's not taken up by an adjacent carbon is going to have a hydrogen attached. Now, unsaturated fats are where some of the bonds between the carbons in that hydrocarbon tail are double bonded to the adjacent carbon. So that means one of each of those carbons bonds are now being joined together with each other. So it'll have two. Each carbon will be sharing two electrons with the other carbon. So it makes a double bond. Okay, those double bonds change the molecular structure. They change the geometry of the molecule itself. And so it makes these little kinks in the hydrocarbon tail. And that's what makes it unsaturated fat. So here's a saturated fat, an example of a saturated fat. What I want to show you something is this is how we kind of do a little bit of our nomenclature for these things. Is we start at the methyl end, the CH3 end here, and we count carbons. So we say this is the one carbon. The one here is the two carbon. That was confusing when I said it like that. This right here is the two carbon. This one is a 3 carbon. This one's a 4 carbon, 5 carbon, 6 carbon, 7 carbon, 8 carbon, 9 carbon, 10 carbon. Okay, so this particular molecule has 10 carbons. You're going to see it drawn like the image on the right. Okay, and the image on the right is just it's a mirror image of the one on the left. It's the same molecule. So take a look at the two structures for a second. See if you can notice some of the similarities. All right, so they're mirror images of each other, but they are the same molecule, right? So basically, molecules are three-dimensional structures. They can be rotated, so if you have a carboxylic acid on this side, it can spin, right? And your point of view changes it to be on this side. And so it's not uncommon to see them drawn like facing a variety of different directions. We'll see that a lot coming up. So anyways, this is the shorthand over here on the right. This is the shorthand method for drawing these molecules. And the way it works is that everywhere you see a point on this line, everywhere you see like it come to a triangle point, there's going to be a carbon there. And also anytime you see an intersection of lines that all come together, there's going to be a carbon there. Anytime you see a line end, like the very end of a line, there'll be a carbon at the very end of the line unless it's specified in any other way, right? And so unless you see lines drawn off to like a circle, right? A circle. Unless you see lines drawn off to an O, like this in this case, it's oxygen, right? It could be drawn off to an S. It could be drawn off to an OH. So anytime that you see lines connected to a letter, right, that represents another atom, right, the center of those lines where they connect is going to be a carbon. And so the way that we count this is if there isn't those lines connecting to those letters, we just assume that the bonds of the carbons are filled up with hydrogen, that they're saturated. And so we can count this and make sure it's the same molecule. So you've got one carbon here at the end, 2, 3, 4, 5, 6, 7, 8, 9, and then 10 right there. Okay, so it's the same molecule. So you're probably going to see me draw them like this for most of the next, whatever, couple of weeks. And it's just easier, but just remember that it comes from this structure. And this structure, all of the bonds make sense. Like the carbons all have 4 bonds. Oxygens have 2 bonds, and that has to do with how many electrons they're sharing with another atom, right? Okay, cool. So moving on to unsaturated fats. So unsaturated fats, like I said, they have kinks in them. And so when we're counting these things, if we're counting back to the first carbon we come to where there's a kink. So I start here and I say 1, 2, 3. This is 3 carbons back, and I have a double bond. I have a kink. And so what I call this an omega-3. So if you've heard people say like, oh, you need to take more omega-3s, well, that's what they're talking about. They're saying like there's a double bond here between that third and the fourth carbon. Pretty interesting. Moving on. So let's talk about so here's the shorthand method, right? But let's talk about some of the properties of these molecules. What's interesting about these molecules is that they all have the same number of carbons in them. Go ahead and count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, right? And then this one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. I'm not going to do the other two, just believe me, right? Okay, so there's 18 carbons in the saturated fat and then 18 carbons in these unsaturated fats, okay? But what I really want to draw your attention to is the melting point, okay? Because what this melting point does is it speaks to what we talked about last time with van der Waal interactions. Van der Waal interactions dictate how similar molecules, right? It governs how they interact with each other, right? So a similar molecule being like, hey, if I have a whole vat of these saturated fats, the stearic acid, right? And they're all hanging out with each other, their molecular geometry, right? Their shape, the molecular geometry and the shape of this molecule is in such a way that if it bumped into another molecule with the same shape, it could get pretty close to it. Like their configurations would line up pretty easily, and they could come in closer proximity to each other. Well, molecules of a similar type, they're attracted to each other. They can dissolve each other, and they can kind of like glued to each other, you know? And that's that van der Waal interactions where you've got two nonpolar fats, and even though they're nonpolar, so that the atoms and the bonds are equally sharing electrons, even though they're nonpolar, they're still both passing their electrons back and forth. And at any given moment, there's going to be a positive charge on one side of the atom or one side of the molecule and a negative on the other side. And same thing for the molecule next to them. And so there's these instantaneous like moments of magnetism between the molecules because they're passing electrons back and forth. And so if you have something like this stearic acid in a vat with other stearic acids in a big vat, they're going to be able to get way closer to each other. And then those forces are going to be much more pronounced. And so they're going to zip together and they're going to be a solid, even if the room is really, really warm. So saturated fats tend to stay solid, even at room temperature. And then the longer the hydrocarbon tail, right, the more gluiness between the molecules and the higher the melting point. So the melting point is just the temperature at which it goes from solid to liquid. So anytime we introduce heat to a system, anytime we add heat to a system, we increase the movement of the molecules of that system. That's all heat is. Heat is movement of molecules in a system. It's the friction of molecules that are moving in a system that are bouncing off each other and rubbing against each other. So that's heat. And so when we add heat to a system, we increase the vibration of the molecules and we shake them apart and they become more liquid the warmer they get, much like butter or something, right? Okay. So having said that, this has a really high melting point, right? 69 degrees Celsius, that's like sauna temperature is where this thing starts to melt. Dry sauna temperature. And then we add a kink in the tail, right? So we have a monounsaturated fat. It's only got one kink in the tail. And notice that one kink drops the temperature at which it will turn from a solid into a liquid. Geez, like almost, what, 50, 60 degrees. Add another kink in the tail, and it drops it by another 20 degrees. And it's like, holy cow. So all these kinks in the tail make it become liquid at a much lower temperature until you get to this point where you have something like olive oil that's a polyunsaturated fat, and its melting point is like below freezing. So you can put this stuff in a freezer and it won't freeze. Something to think about. And it's because of those kinks in the hydrocarbon tail. Sorry, my dog's barking. Give me a second. I'm just going to pause this and then tell him to be quiet. All right, and we're back. So we talked about what causes the different melting points of the fatty acids. Go ahead and answer that question real quick in your head. It's a different, I'm phrasing it slightly different here. What is causing the different melting points? It's the kinks in the hydrocarbon tails, right? Okay. It's the double bonds between the carbons in the hydrocarbon tail. And so here's an example. Let's take a look at this. So all these are fatty acids. You've got the carboxylic acid group is on the far right. Methyl group at the very end is here on the left. And so what this is showing you okay, here's a saturated fat, okay, and we'll call it palmitic acid. Here's an omega-9 saturated fat. So we know that because we can go and count from the methyl end, count 1, 2, 3, 4, 5, 6, 7, 8, 9 carbons back, and I get to a double bond. So that's an omega-9. And then if I want an omega-6 9, right, I count 6 carbons back and there's a double bond. Keep counting, 9 carbons, that's a double bond. So that's an omega-6 9 fatty acid. And then you've got down here, you've got omega-3 6 9 fatty acid. So that's give some of the nomenclature to like omega fatty acids and stuff. Now, just a fun like nutritional fact, right? Because I'm gonna want you guys to go out and get a nutrition label, take a picture of it, upload it to Canvas. Fun fact about fatty acids though, and omega-3s, and just sort of like where did that come from? Like where did this whole like, oh, take omega-3s and your heart will love you and it's good for your heart health and decreases inflammation. I'm sure many of you have heard these adages, and it's like, where does this come from? Well, so historically, ancestrally, long before like we had industrialized farming and stuff, the diet, the ratio of omega-6s to omega-3 fatty acids. So that would be like an omega-6 would be 6 carbons from the tail, right? That would be a double bond. And then that would be the only double bond. So it'd be a monounsaturated fat. Then you have 3 carbons in, right? And so that's another monounsaturated fat. But the ratio in our diet of those fats to each other was we had for every 4 omega-6s, we were getting 1 omega-3 fatty acid. And that was like the proportion that we wanted in our diet. That's what our bodies were adapted to. Along comes modern agriculture and the corn industry, right? Goes through there. So you got corn oil, corn syrup, all this stuff, right? It is corn based. Well, corn is entirely omega-6 fatty acid, like in its cell membranes. And so what that's done to our diet is it slowly raised the amount of omega-6 that we're getting to the omega-3s. And so because of all this corn in our diet, our omega-6s are going up really fast. And so we're actually at a 20 grams of omega-6 to 1 gram of omega-3 is the average American diet, the average Western civilized diet, or whatever you want to call it, right? So anyways, the ratio has climbed. It's 24 to 1 right now, unless you're supplementing. And so people will take omega-3 fatty acids and supplement with them to boost the amount of omega-3s they're getting per omega-6. And so that's just where that comes from. Anyways, I digress, completely digress. Let's talk about making a fat molecule. So making a polymer from the monomers, right? In this case, so whenever we make a polymer of a fat, we're using a glycerol backbone. Now, glycerol is actually a carbohydrate backbone, and I know it seems kind of weird that we're hijacking another molecule from another family to make a polymer with it, but it just so happens that that's just how fats are attached to things. Like in nature, they're usually attached to this small carbohydrate molecule, and that's what makes them into a polymer. So anyways, this is a glycerol molecule. A dehydration reaction is going to link this fatty acid's carboxylic head to one of the OH or the hydroxyl groups from this glycerol molecule. And so a water molecule is going to pop off. And what that does is it frees up the bond on oxygen, frees up the bond on carbon to join with each other. So instead of oxygen being bonded to hydrogen and instead of carbon being bonded to oxygen, those pop off, and then carbon and this oxygen from glycerol make a pact, and they're like, hey, let's play with our action figures together. And boom, there you go. You've got the start to a polymer. So if you do this 3 times, right, what you end up with is triglyceride, right? If you do this 2 times, you end up, oh my gosh, my cat is climbing on me. Okay, come on. Okay. So if you do this 2 times, you end up with a phospholipid. So do it 3 times, 3 dehydrations, you end up with a triglyceride, and that's what you get measured in your blood levels when you get blood work done. Do it twice, boom, boom. So here's 2 fatty acids hanging down from a glycerol molecule. Do it twice, and then attach a phosphate to that third one. So instead of 3 fatty acids, this phospholipid is going to have 2 fatty acids, and then this third carbon up here from glycerol is going to have a phosphate group. Oh my gosh, kitty, stop it. Okay, you're done. Okay, and then just ignore this stuff on top. That's just like this stuff makes up cell membranes, and so like attached to this phosphate group is going to be like any number of things, like consider you're a cell and you want to like communicate with another cell. Well, you have to have like antennas, receptors. You got to be able to like send molecules and stuff. And so these things sticking off of the phosphate group are varied. They can be just about anything. Let's see. So where were we? Phosphates. So this is a phospholipid, and it's just one half of a cell membrane. A cell membrane is a phospholipid bilayer. So it's got two sets of phospholipids, and they face each other. Because remember, like dissolves like, and so these hydrocarbon tails are completely nonpolar. It's a fat, right? It's a fatty acid, so it's not going to dissolve in water. But if you have two sets of them, the two sets of fatty acid tails will dissolve into each other. And then what sticks out into solution, what sticks out both into the inside and outside of the cell, are these polar phosphate heads. And water is polar, it's a polar bond, and then this phosphate group is a polar bond. And so there's a hydrophilic and a hydrophobic end to these things. So there's a hydrophilic end where the phosphate group is, and a hydrophobic tail. Okay, and so this is an example of a phospholipid bilayer. There's an inside and an outside. It depends on how you look at it, right? So there could be an inside to the cell here. There's an outside to the cell here. And the two sets of phospholipids come together to make the phospholipid bilayer. So here's just sort of a graphic image of the molecule itself. And you'll see that the tails, the hydrocarbon tails, dissolve into each other. And then notice they drew a bunch of water molecules on the outside of the cell to show that they have this affinity for the phosphate group. The phosphate group, because it has a lot of oxygens, is incredibly negatively charged. And if you look closely in this image, the illustrators were just paying attention enough to turn these water molecules so that the hydrogen ends, which would have a more positive charge, right, are facing this negative phosphate group on our phospholipid. And so they're doing that because they're attracted like magnets. Crazy. Okay. So anyways, here's an artistic representation of how a cell membrane might actually look. You see the phospholipid bilayer, right? You see how these fatty acid tails are kind of like, or hydrocarbon tails are dissolved into each other, and the phosphate heads are facing the water on the outside and the water on the inside of the cell. You also see these giant like oblique looking I don't know, they're just blobs that are sticking into the membrane. Well, these things are transmembrane proteins. So that's a whole nother class of biological molecule. So these are transmembrane proteins. They do a lot. They can pump stuff in and out of the cell. They can be a channel for like ions to flow through. They can be a signal thing. So it could be like a signal comes in and binds to it and then changes the conformation of this protein. And then that signal is transduced into the inside of the cell. So it's sort of like if somebody like gave like Morse code tapping on your door and you wrote it down on the inside and then gave that message to somebody. Like the message didn't actually like come through the door. It was just like transmitted through the door, transduced through the door. Okay. What you see here in the membrane, right? This is not an accident. There's this structure here that we're going to focus on, and this is cholesterol. So this must be an animal cell membrane because it's got cholesterol embedded into the phospholipid bilayer. Out here, this is just like extracellular matrix. It's on the outside of the cell. It's part of the signaling, the structure of the cell. It's part of the filtration system of the cell. I mean, I'm not saying these things are filtering anything, but it filters what comes in. It's a selectively permeable membrane. And sometimes it lets stuff in, sometimes it doesn't let stuff in. And then on the inside here, this twisted rope looking stuff, that's the cytoskeleton. So that also made out of protein and gives structure and form and definition to the cell itself. So let's focus on this. So we're going to focus on the cholesterol next. So cholesterol is another lipid. Okay, you'll see it has a hydrocarbon tail here. Boop, 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 boop. And then it's got these hydrocarbon rings. Okay, don't need to know what these are, right? But you'll see them a lot in chemistry. You'll see them a lot in biology. They're just rings of carbons. And then, you got a little polar group out here. But for the most part, this molecule is pretty much non-polar. Okay. This OH group might attach to like something on the outside of the cell too. Okay. So what are the functions of cholesterol? Well, we know, we've heard this before, that cholesterol is bad for you. Don't eat too much cholesterol. It'll raise your blood something, I don't know, may give you heart disease, right? But cholesterol is super important to the body, especially animal bodies, especially animal cells. So what it does for animal cells is that it's embedded into the phospholipid bilayer. And the phospholipid bilayer is as tender as a bubble, right? So putting in this cholesterol helps to kind of stabilize it, but it also allows it to have some flexibility. So it keeps its shape, but it also kind of bends it. So think about like cholesterol is kind of like the material that Gumby's made out of. It can bend and flex, but then it'll come back to its original shape. And that's sort of like what cholesterol does for animal membranes, which makes sense because we want to be able to move. Whereas something like a plant needs to withstand like a hurricane. So it needs a cell wall. It needs an additional layer on the outside of it to keep it supported. Whereas we don't need to be as stiff as a plant. So we need that fluidity from the cholesterol in our membranes. And that's also why when you eat like steak or you eat animal products, they came from animals, so they have animal cells in them and they got cholesterol in them. So you cannot eat animal products and not get also some cholesterol. So that's where that comes from. But the importance of cholesterol inside the body, inside our system, is that it's used as a precursor for lipid-based hormones. And so lipid-based hormones are the ones that go right through cell membranes, because remember, cell membranes are like soap bubbles. They've got those hydrocarbon tails in the middle that are nonpolar, that are a lipid. They're nonpolar, right? And so you have these lipid-based hormones that are based out of cholesterol that can just dissolve right through cell membranes, dissolve right through the nucleus, and go right to the DNA and start switching on your genes. And so this might look familiar: testosterone, estrogen, right? These are hormones of like endocrine development, hormones of maturation. These are hormones that directly impact the DNA. Most other hormones just interact with the outside of the cell and they just kind of like, hey, you got a message, and then the message gets transduced. These ones don't care these ones don't care if there's a messenger at the gate. They just go right through the gate and then straight to the king and change the system completely. You got progesterone, aldosterone. These ones are in the kidneys. They help to regulate like salts, like how much salt is in your body, stuff like that. Cortisol, like a stress hormone, right? Again, targets every cell in the body. Okay, so that's the functions of cholesterol. We're going to move into carbohydrates. So many of you are familiar with carbohydrates. This is the next portion of a nutrition label. So you've got the fats, right? And we want that for the assignment for the discussion board. Want it to have different types of fat, then carbohydrates. And so if you look under a carbohydrate label or under carbohydrate on a nutrition label, you'll see that it's separated into several things. And a lot of times, I think nowadays there's been some legislation passed where it tells how much sugar is added typically now. So how much sugar do they add on top of the natural sugars already occurring inside of whatever this package is you're buying? Okay, so you've got simple sugars, and then you've got complex sugars, and that's about as far as it needs to go. Simple sugars would be the monomer forms of the complex ones, right? So the monomers will build together to make complex carbohydrates. And so we'll talk about where those come from in a little bit. Let's go. So carbohydrates are also called saccharides. They end in the suffix ose, os, right? So glucose, fructose, maltose, sucrose. It's characterized by the formula for every carbon. So I can substitute a number for n for n. So if I put in a 5 for carbon, then I'm going to get 10 hydrogen and 5 oxygen. So for every 5 carbons, I get 5 water molecules, right? Because 5 water molecules will have 10 hydrogens and 5 oxygens in them. So anyways, that's where we get carbohydrates, the water portion. So it's characterized by that formula. They don't always obey that formula, but that's typically how we see carbohydrates. So simple sugars or the complex carbohydrate, which is the polymer form. Here are some of the monomer subunits, right, of the carbohydrates. Don't freak out. Here they are. Okay. So you've got triose, so that's 3 carbon sugars. You've got tetroses, that's 4 carbon sugars, pentoses, and hexoses. If that looks like a lot, believe me, it is, right? This isn't an organic chemistry class. So let's simplify it. I want you to know these ones: glucose, fructose, ribose, deoxyribose. And by know what they are, I don't mean like you look at it and you're like, oh yeah, that's obviously a glucose. No, what I want you to know is like be familiar with the term glucose, right? Know that it's a simple sugar and know that it's the monomer of a polymer of the sugar, or a monomer of a starch or a polysaccharide. Okay, so we've got glucose, fructose. Glucose and fructose actually have the same chemical formula. They're both C6H12O6. We can count the carbons. Look, 1, 2, 3, 4, 5, 6. So in the cases of these points here, they're not actually drawn as the letter C. This is the shorthand method, but up here it is, right? And so we've got 6 carbons here, and we've got 6 carbons here. 1 here, that's the C. 2 here, 3, 4, 5, and 6. So they're both C6H12O6. So if they have the same chemical formula, right, why are they different things? Why are they called different things? They're isomers. Okay. They're isomers because they're built out of the same building blocks, but their overall structure is different. So if I handed you a bag of Legos and you a bag of Legos, and I said, build me something out of all these and use every single one, they would have the same materials, but your structures would be completely different, likely more different than even glucose and fructose. And then, let's go back for a second. Ribose and deoxyribose are the 2 sugar molecules used in nucleic acids. Okay, nucleic acids are a little more complex than this, but this is like the central part of a nucleic acid. And so this is where we get DNA, that's the D from DNA, and this is where we get RNA from, which is the R in RNA. And you'll notice the only difference between these two is that deoxyribose does not have an oxygen here, and ribose does have an oxygen there. That's it. Yeah. So quick review question. If we're going from a monomer to a polymer, if we're building from a monomer to a polymer, right? What type of reaction is this? So it's a building reaction. So we know it's anabolic. What's the E word? Endergonic. 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 So that means it requires energy to come in from an outside source. So we're going from monomer to polymer. We're doing an endergonic reaction. And we're also doing a dehydration reaction. So we're going to take an OH group from one glucose, an H group from the other, well, just a hydrogen, it's not even a group, a hydrogen from the other glucose, dehydrate the molecules. The water is removed, and that frees up a bond on one of the glucose molecules and frees up a bond on the other glucose, and then they share electrons across that bond. And so what that does is that makes maltose. And so we have a disaccharide here, a 2-saccharide, di meaning 2, and then saccharide meaning sugar. So there's a disaccharide. And then another dehydration reaction in the synthesis of sucrose, which is table sugar, right? You can use an enzyme and actually do this reaction in reverse and take table sugar, sucrose, and you can actually digest that bond and turn it into a syrup that is made up of fructose and glucose, and that's called high fructose corn syrup. So that's all they do is they take an enzyme, they break this bond here, and they go backwards, and they separate it. And they say, you know what, we don't need this linkage here. We don't need that bond between the two. And so that is how you get high fructose corn syrup. This is how you go in the opposite direction. You take glucose and fructose, you dehydrate them, right? The bonds, that bond frees up, and then they link together here. I know there's a lot of letters going on this page, right? And like arrows and stuff like that. So over here, this is just one side of the equation. And when it gets dehydrated, it turns into this side of the equation. That's what you're looking at. Okay, so the first type of polymer that we're going to come across. So this is just how it happens, right? It's dehydration is how we make a polymer. But the first type of polymer that we're going to talk about is glycogen. So glycogen is unused glucose that is in the bloodstream or in the cell, okay? So muscle cells, because they require a lot of ATP to be moved, and we're not just talking about exercise, we're just talking about, hey, I need a drink of water, I pick up my glass. That requires muscle activity. That uses up ATP. So muscles are going to need a lot of ATP to do their job, to just move as a body, right? Exercises especially. But the extra glucose that they have, they're going to store it as glycogen. So muscle tissue has a lot of glycogen, but so does liver tissue. So liver tissue, if it receives a signal from your pancreas that's like, hey, you know, I got a lot of blood sugar going on here, can you ramp up the glycogen production? The liver will take in the glucose molecules and convert it into glycogen. Okay. So we'll touch base on a couple of these, but the summary of carbohydrates are the 4 that I want you to know are glucose, fructose, ribose, deoxyribose. The polymers that I want you to know are glycogen, amylose, and cellulose. And you might say, wait a second, we haven't talked about amylose or cellulose. Well, let's talk about them now. Cellulose is used only in plants. Okay? It's not the stuff that's on your thighs, right? It's the stuff that's in plants. And it's in the cell wall of plants. So when plants have some extra sugar that they need to store, they might make a new cell and they might ramp up the production of their cell wall tissues. And that's made out of cellulose. Cellulose is dietary fiber. So when you're looking on a nutrition label, right, and you're like, oh, look, it says my dietary fiber. That's cellulose. Cellulose is undigestible. You don't have the enzymes in your body to actually break that stuff down, so it becomes dietary fiber, creates a bolus, pushes it out, you got feces. Okay, good story. Anyways, so that's cellulose. The other type of starch, the other type of carbohydrate polymer, or the other type of complex carb, right? There's a bunch of different names for them. The other type of polymer is made inside of the cell. So it's made inside of the plant cell. And the plant takes extra glucose and it bonds them together and it makes starch called amylose. And so that's the starch that you find in like crackers and flour. That's the starch that you find in potatoes and that kind of stuff. So that's the amylose starch. Amylose, complex carbohydrate, also called a starch, also called a polysaccharide. So lots of different names for those. Okay, let's talk about proteins. The monomer of a protein is an amino acid. And so you probably heard like, take your amino. I need some protein in my diet. I need to gain muscle, or I have some branched chain amino acids. They help me with muscle recovery and that kind of stuff. Well, yeah. So branched chain amino acids are just you're ingesting a whole bunch of monomer subunits so that you can repair your muscle tissue, and your muscle tissue is polymers. So you've got the monomers and branched chain amino acids, and they can help to build the polymer, which is your muscle tissue. And so proteins, we typically think about them in terms of like muscles, or I need more energy, right? Like I need some energy that's not going to spike my blood sugar. Well, there you go. That's protein, right? So that's just part of the story, right? Proteins are actually literally what makes everything in a cell function, makes everything in a cell operate, are all by proteins. So you can think about like proteins are the doers, they are the builders, they are the breaker downers. Okay, they're the doers, the builders, the breaker downers. Okay, they also have structural components. So there's, proteins build up the cytoskeletal elements inside a cell. The extracellular matrix are made up of proteins and carbohydrates, some of those too, right? Everything's kind of mixed in there. But proteins are primarily the doers in terms of enzymatic reactions. So they're the enzymes. They are the breaker downers and the builder uppers. Okay. All right, but let's see. How does that actually work? Well, let's take a look at the monomer subunit. We'll talk about its characteristics here. So you've got a central carbon, right? And then attached to that central carbon, you have three important units. You've got this carboxylic group over here, and you've got this amine group over there. I don't know how to get rid of that thing without, every time I move over there, and I don't want to hit exit because that'll exit the show. Anyways, you've got an amine group over here, carboxylic group over here, and then this functional group here, right? So central carbon bonded to two functional groups that are always going to be on that central carbon for every amino acid monomer, right? But then every amino acid monomer is going to have an additional functional group that gives it its identity. So you can think about like this structure down here, everything except for the R is sort of like the shell, right? Or the frame of a car. And then the R is like everything that goes into the frame of the car. So you've got like this is your car frame, right? This is the car frame. You could attach like a Ferrari engine to it, or you could attach like a go-kart engine, or you could attach like three wheels to it or something, so that you can attach different things to this shell of a car, but the shell is always the same. Okay, and so that's how it works. So we have about 20 different things that we can plug in to this car frame, if you will, right? That changes its function, changes its identity. If you put a Ferrari in a car frame, it's going to be a Ferrari, right? If you put a go-kart in a car frame, it's going to be a go-kart. It's not going to go very fast. But anyways, there's 20 different things that we can plug into this molecule. Okay, so don't freak out, right? We're going from this image here to this image up here. And the only thing that's changed is that this R group now, I'm showing you all of the mods, right? All of the attachments that can come into this car frame. Okay, so the attachments are all shown in yellow, right? That's the functional group, or the side chain is sometimes what they're referred to. The functional group or the side chains, right, are in all of these colored boxes. And so notice the frame of the car is always the same, right? The frame of the car is that purple box. Okay. Something else you might have noticed, right, is here I've got an H, and over here I just have NH2, right? And as we come up here, we lose the H over here, so that's that hydrogen proton popping off. And if you remember, nitrogen has an extra lone pair of electrons, and so that hydrogen proton is pissed off. He's looking for some electrons to like make himself feel better. And he comes over here and he's like, "Oh, nitrogen's got a lone pair he's not playing with," and it comes and joins the nitrogen. And so that's what makes the amino part more basic because it pulls the proton out of solution, and that's what makes the carboxylic part more acidic because it drops the proton. So this is actually like it's like a yin and yang like going on right here. It's kind of cool. So you've got yin and yang, and then attached to yin and yang, you've got your functional groups and your mods that you can do to each one of these amino acids. Now these mods, these amino acid mods, or these amino acid functional groups, they they have characteristics that belong in general groups. So the non-polar mods are all up here, and so those are water-fearing. When these appear in a polymer, they tend to turn inward towards the inside and not face outward towards where the water is in the cell, right? You also have polar side chains or polar mods, and these ones are water-loving, and so they typically end up facing out when you get a polymer of an amino acid. And then you've got acidic ones that drop protons, and you've got basic ones that pick up protons. So you'll notice the acidic ones are carboxylic based, right? And the ones that pick up protons that are basic are amino based. And so that's kind of interesting. Okay, so moving on, let's talk about how we make a polymer out of these, right? So I want you to think about it like this, right? So I'm going to show you this image. We'll come back to it, but I want to show you it like this first. Okay, you can think about amino acids in polymer form as being beads on a string. So you have a string and you just put beads on that string, right? And so if I was to ask you what is the significance of the beads on that string, you could say, well, it's the order in which they come. It's the pattern in which they make, right? So unfortunately, this author here should have, if he was doing it right, colored all of these circles in with a color, because the color gives like some identity to that amino acid, right? The color represents the functional group. It represents the characteristic of that amino acid. And it's if I say, okay, why did you put all of these beads on this string in this order? Well, if all of the beads have a different, size and a different color and a different look to them, right? You would organize them on that string. You would organize them on that necklace, that bead, in a certain pattern that makes sense to you, right? And that pattern might be like, oh, red, blue, purple, spiky one, red, blue, purple, spiky one, or something. And then maybe you change the pattern a little bit. And it's the same thing here. Amino acids are just beads on a string. But here's the crazy part, right? And that the crazy part is that molecules are magnets. And so each one of these amino acids is going to behave either like a non-polar or a polar molecule. So the nonpolar amino acids, the nonpolar ones that have nonpolar side chains, those are all going to try to find each other. And so you get this yeah, it's a two-dimensional string and beads on a string, but certain beads are attracted to each other if they're nonpolar, like dissolves like. Certain beads are attracted to each other if they're polar, like dissolves like. And so it ends up twisting this string into a complex 3D shape based on the functional groups of each amino acid and how they interact together. Now, this is primary structure, what I showed you here, and I'm going to show you what comes after that. But let's go back to how we actually make a polymer. Monopolymer is a dehydration reaction, right? I can't stress that enough. You lose an OH group from the carboxyl end of the amino acid, and you lose a hydrogen from the amino end of the next amino acid. So this is the growing peptide, polypeptide. We're adding this new amino acid to it, right? We dehydrate both, and then there's a free bond between carbon and nitrogen, carbon of the one amino acid, nitrogen of the next one. And that bond that forms is called a peptide bond. And so what we've done is we've gone from 2 amino acids in a polypeptide to 3 amino acids in a polypeptide. And so what, if you'll notice, if we keep adding on, right, if we keep adding on amino acids, all of the amino acids are going to be oriented in the same direction, right? The amino group is going to be on one end, And since the carboxyl group is bonding to the amino end on the next one, right, there's a directionality to the growing string of of amino acids, right? There's a directionality. One side of it, one head of it, if you will be an amino head. One side of it will be a carboxylic tail. And that's what we see here, right? So we have the amino head of the growing polypeptide is here. And we're just tacking on, making peptide bonds, right? Making this huge beaded string with magnetic molecules on it. And then boom, we get to the carboxylic end. So now this primary structure can actually fold into a variety of secondary structures. One of those secondary structures is an alpha helix. And so if you remember, right, we made beads on a string, and some of those beads are magnetic, some of them are attracted to each other. And there's a pattern to how they appear on the string. And so if it's red, green, blue, purple, spiky thing, red, green, blue, purple, spiky thing, what those things can do is then they can start to twist into a spiral where maybe the spiky thing is interacting with the red thing and attracted to it. And so it twists up your beads on a string to kind of look like a slinky, right? And that would be an alpha helix. Another pattern that sometimes these repeating amino acids do is they fold into a sheet, kind of like a serpentine sheet, where it goes down one way, drops down, comes across the other way, down and across. And so this is the secondary structure of a polypeptide. Now keep in mind that, like, you've got beads on a string. Not every single bead on that string is going to be part of one of these helixes or pleated sheets. Sometimes they'll just remain in their kind of loopy, like stringy state. And so it's really it's the secondary structure interacting with elements from the primary structure, which is the beads on a string, that form the tertiary structure, right, which is the three-dimensional shape of the protein. And so it goes primary, secondary, tertiary. And tertiary, as you can see here, you've got part of it that's an alpha helix, part of it that's that serpentine beta pleated sheet, but other parts of it are just stringy primary structure, beads on a string elements. Now, if you have more than one tertiary structure, like hemoglobin, where it's got multiple subunits, this protein structure here, this three-dimensional structure, is just one piece of a bigger robot, right? So it's one piece, like it's like the arm cannon of a robot, you know, and it needs to join to like the chest piece. And so you can build these large quaternary structures that have multiple subunits attached. And so that's why proteins are so fantastic, is that you can take from very simple things like simple amino acid monomers and build these incredibly complex machines at a microscopic level that can do amazing things in the body. Okay, let's talk about nucleic acids. All right. So the monomer of a nucleic acid is called a nucleotide. Okay, so a nucleotide can be either DNA or RNA. There are RNA nucleotides and there are DNA nucleotides. How we differentiate what is RNA and what is DNA is based on the central sugar of each nucleotide, right? So a nucleotide is made up of three components. It's made up of a ribose sugar, right? And ribose is a pentose sugar. It has 5 carbons. It's made up of a ribose sugar. It's made up of a nitrogenous base, of which any of these up in this box here could be substituted for that this hexagon right here. So you could have adenine plugged in there, guanine plugged in there, thymine plugged in there, cytosine plugged in there. If it's RNA, it would be uracil, and we'll talk about that in a second. So 3 different components. There's a nitrogenous base component. There's a pentose sugar like ribose or deoxyribose, and then there's a phosphate group that's attached to the 5th carbon on that ribose sugar. That phosphate group is what links one nucleotide to the next nucleotide. And so the phosphate group from an incoming nucleotide, so let's say like this nucleotide is joining to this nucleotide to become a polymer. The phosphate group of this nucleotide that's attached to that 5 carbon is going to dehydrate with the OH group on the 3' carbon from the one above it, and it's going to dehydrate that reaction, and then it's going to link up to the 3' carbon on the next one. And the 3' carbon is right here this is on the deoxyribose, and this is on the ribose sugar. I know that's a lot to take in, and that's nothing that I really need you to like regurgitate on a test, but it is something that you're going to see again if you're in a biological sciences major, right? DNA polymerization is like fundamental to like most biology classes. And so that was just like a super fast explanation of how it works. So dehydration reactions at the level of the phosphate group attaches the phosphate group between nucleotides, right? And so that's why we say that DNA has a phosphate backbone. It has a negative charge. DNA has an overall negative charge because those phosphate groups attract a lot of electrons, right? So DNA is a very polar molecule. It will, if you put it in current, if you drop DNA in current in like a, if you run direct current through like a water tank, right? A negative end and a positive end, DNA will run right to the positive end. It's attracted to it like a magnet, right? Okay, I digress a little bit. The sugars. So the difference between DNA and RNA really comes at this 2' carbon position. And it's that RNA, the ribose sugar, has an OH group. So RNA is able to participate in dehydration reactions and also in enzymatic reactions. And so RNA is less stable. Like more things want to interact with it at that 2' carbon level. Whereas DNA, it doesn't have that OH group. It just has a hydrogen there. And so it's less reactive. And so this is why DNA will stick around on like fibers of clothes for years, but RNA will be gone in a couple of hours, right? DNA is much longer lasting because see all its OH groups? They're all taken up with things. This OH group up here has a phosphate group attached. This one has a nitrogenous base attached. And then the one down here is attached to the phosphate group of the next DNA or the next DNA nucleotide in the strand, in the polymer. And so if all the OH groups are taken up, that means there's nowhere to react with it. And so DNA is incredibly stable, which is good because you don't want your DNA getting all jacked up, right? Okay. DNA is typically double helix. RNA is typically single-stranded. Typically, RNA exists in many forms. So we can think about DNA as being sort of a repository of information. Okay, there's an order and sequence to the strand of DNA, right? That is made up and constituted by the nitrogenous bases that are part of each nucleotide. And so if you were to look at a strand of DNA, right, this one has looks like adenine, this one might be cytosine, maybe guanine, followed by another cytosine or something, right? So there's a directionality to DNA, right? There's an order and sequence to which its nucleotides appear. There is an order and sequence to which the nucleotides are attached to each other in this strand. There's an order and sequence to which the nucleotides are placed in order here on the RNA strand. And here's the cool thing, is that the order and sequence that those nucleotides appear is paralleled to the order and sequence that amino acids appear like beads on a string. Think about that. Might say big deal. Like, what does that mean? Well, think about this, right? We talked about proteins ad nauseum for a little bit ago, and the protein's character, like its 3D structure, is dependent upon the order and sequence of those beads on the string and how they interact with each other. And the order and sequence of those beads on the string is parallel to the order and sequence of nucleotides on a DNA molecule. And that's why DNA is considered a blueprint, right? Is that it tells us what the order and sequence should be on an amino acid or on a polypeptide, on a protein. Okay, so anyways, the only real difference between RNA and DNA is that RNA is less stable, DNA is more stable. RNA is a chemical messenger, but it can also be many other things. And we'll spend almost all of Unit 3 talking about the different functions of DNA and how it works, right? But here's the major differences. DNA is more stable, RNA is more transient. RNA uses uracil as a base, as a nitrogenous base in its nucleotides, whereas DNA uses thymine. And if you look, the only difference between thymine and uracil is a methyl group. That says H3C or CH3, right? There's 3 hydrogens and then 1 carbon. So there's a methyl group on thymine and there's not a methyl group on uracil. That's it. Other than that, the molecules they use are exactly the same. Literally, like they look exactly the same. DNA is also missing that OH, right here. It's just missing that the oxygen from its 2' carbon. And that is literally the only difference between DNA and RNA, molecularly, structurally. Cool. So that's the end of nucleic acids. Let's give a little summary and then we'll end this lecture. So DNA, deoxyribonucleic acid, stores information on how to build a protein or RNA molecule. So remember, RNA comes from DNA. RNA is a photocopy of DNA, okay? It's missing the OH group. It's more stable. It's double-stranded. RNA is usually single-stranded, and it's a direct RNA copy of the DNA, and it can be functional and structural. We'll talk about that in the next in Unit 3. and it can be turned into a protein. Like I said, it's parallel. The order and sequence of nucleotides in an RNA strand is parallel by the order and sequence of amino acids on a polypeptide. So it's pretty interesting stuff. And there's lots of different types of RNA. That's all for Unit 3, though. So that's the end of the lecture. Hope you guys enjoyed it. I'll catch you later. Bye.